record just so you can watch this later. So like, okay. I can't minimize, so what do I do? Just go here. Does this look familiar?
No.
Does that look familiar?
Yeah.
All right, so let's just pick up. These are the shapes. It's a common shapes of bacteria. When it comes down to it, bacteria, all bacteria, would be one of these three shapes, right? And you can pretty quickly identify these guys. Bacteria, again, are very, very tiny, incredibly tiny. But if you have a good microscope, you can see them. And one of the first things to look for is their definitive shape. And so here we've got common names and we've got official names. I think you've certainly heard of some of these official names before. They sound kind of strange, actually, but again, back to Greek particularly. Anyway, here we go. What are the shapes? The first one is simply spherical, round. Many bacteria are round, and you can see that in the illustration here. These are the typical round shaped spherical bacteria. Now, the real name for that is coccus or cocci. Now, some of you have gone to the doctor before, and they've done a test, let's say, on your throat. They've swabbed your throat, and maybe they've come back and said you have what? Strepto what? Yeah, you may just say strep, and that's probably what they say, but the official is streptococcus. In other words, those little bacteria that are occupying your respiratory canal. Yeah, specifically, it's a type of strep, but more specifically, the shape of those cells is spherical. That's the streptococcus. Okay, the other would be rod-shaped, or you say rice grain, kind of like the typical rice grain shape structure. More officially, that is called bacilli or bacillus. And then you have this third shape, which is actually very rare. Probably less than 5% of the bacteria would fit into this category. This is basically a little longer rod-shaped cell, but the key here is it's spiral. So if you see one that is spiral, that is called spirilla, or sometimes more commonly spirochetes. So again, you got the spherical coccus or cocci, you got the rice grain shape, rod shape, bacillus or bacilli, and then you've got the curly ones, right, the twisted ones, those are spirilla. Any of those, do those names sound familiar from life science? Spirilla. One of the students yesterday said that y'all did an activity in 7th grade, something about, I forget the title of it, something about something spirilla.
I think so.
Does that sound?
Yeah, baseball.
Forgive me and tell me if I've shared this story, because I can't remember what I shared before Christmas break. But studying at the university, right, becoming a biology major, took many specific courses, and that's what you do when you're studying to be a biology major. You have a whole semester course that is focused in one area. And so I had one course that was called microbiology. The whole semester course was nothing but microorganisms, and particularly bacteria. A lot of the course was just bacterial cells. Does this sound familiar?
Yeah.
Yes, I told this.
I don't know.
I mean, even yes, I was like,
Okay. So, well, you may first say, Ms. McCaskill, a whole semester course of nothing but bacteria, that sounds like torture. Well, actually, it was one of the most interesting courses that I've ever taken. And I think one of the interest of it was the way the professor handled the labs. And maybe you're thinking at this point, I know still in the distant future, but you're already kind of thinking about college and university. One of the typical ways they structure university, at least for science courses, is you'll have, like let's say a typical week, you might have 3 classes. 2 of those classes would be nothing but lecture, and 1 would be a lab, or something like that, right? In other words, the lab time is separate from the lecture time. Different universities structure things differently, but that's somewhat typical. Anyway, at the university I was at, that was kind of the form. And so we had the first lab of the year, of the semester, of this microbiology class. And the professor came into the class and he had a bunch of test tubes. And in those test tubes was liquid. And basically what he did is he just gave each of the students in the class a different test tube. So he'd be like, Maria, here's your test tube. Be careful. Living bacteria in there, okay? Reagan, here's your test tube. You get the idea. Reese, here's your test tube. Everybody gets a test tube. Now, in the liquid, in the test tube, were bacterial cells, bacterial colonies. Here's the challenge, he said. This is your task the remainder of the semester. First, you now must keep those bacteria alive. They're yours. Okay? Secondly, by the end of the semester, you need to properly identify those bacterial cells that I gave you. How will you do that? Well, I'm going to teach you through the semester things about bacteria, and you will then come to the lab on your own time, and you will apply those procedures that I teach you to figure out which bacteria colonies you have. So it's kind of like a bacterial scavenger hunt. Does that sound? I wish I could do that here. That just takes so much time, because actually, if I think back to that time frame, I was going into the lab frequently, doing different tests and procedures, trying to figure this out. It wasn't something that I could figure out very quickly. It took time, but it was fun. I thought it was fun. It's like a scavenger hunt. One last thing, and here's your lesson for this year, right? As we start a new semester. The professor said one last thing. He said, here's what you can do. You can come to me before the end of the semester. You can hand me a little piece of paper, write the name of the bacteria that you think that you have in your test tube, and I will simply do this or this. Of course, if you get this, good sign, right? You've done everything well. You've properly identified your bacteria, and now all you have left to do is to write a paper describing the procedures you went through and how you properly identified the bacteria. That's all you got left. That's not a big deal, right? So I was not always, to be honest, I was not always the most beautiful student. I mean, I was a good student. I worked hard. I wasn't, I did as a college student, especially sometimes procrastinate. That's always a problem for college students, and I was not exempt from that. But at least in this case, I did pretty well. I worked hard, and I got to the professor early, and the professor did this to me, which was like, whew, right? I mean, just now everything's, all I got to do is write a paper. Here's a lesson. I had some friends who weren't quite so dutiful. They procrastinated a little bit more than I did. And they then brought their guesses to the professor with only like a week left in the semester. And for some of my friends, not all of them, they got this. Which means now they only have a week left to go back into the lab, figure out where they went wrong, and then hopefully identify the bacteria and write a paper. So here's the lesson. Don't procrastinate, right? Get your work done quick, and it'll help you. It'll ease your concerns. And okay. Anyway, that was fun. Now, again, I wish I could do something like that here. I've never been able to do something like that in my biology classes because it just takes too much time. But it was fun. Does that sound fun to you?
Yeah. Unless you don't figure out what you're doing.
Yeah. See, that's the thing is you got to get moving quick and not waste time, and then get your answer to the professor so that if you do mess up, you have plenty of time to figure it out. So, well, why I brought up that story is because one of the first things I did when I looked at the bacteria that the professor gave me, one of the first things I did was what? Figure out the shape. Because if my bacteria was coccus, then I can eliminate all the bacillus, right? I can eliminate all the spirilla. And now I've super narrowed my focus. And that's what you do. All right, just a little bit about how bacteria grow and divide. We're not getting into technicalities here with the process, but the process is, there's a different name here, as you see. Bacteria divide by a process known as binary fission. Bi means how many? Two. Okay. Fission just means splitting apart, right? So binary, two-way fission, splitting apart. In other words, one bacterial cell divides in half. That's basically binary fission. Here's the important question I want you to consider. Why don't we just say that bacterial cells use the process of mitosis? Why can't we say that?
Because mitosis basically split, like, they copy, like, the chromosome of, like, the mother cell and split it into, so, like, each of the daughter cells have, like, the same amount of chromosome. But then bacteria just, like, split the chromosomes. So
Okay.
they only have, like, half of the chromosome of the nucleus cell.
Are you saying for bacteria?
Yeah.
Okay. All right. You're heading in the right direction. There's kind of a piece there maybe to be added to it, but you're certainly heading in the right direction. Any other thoughts here? Okay, so if you think about mitosis, right, we talked about mitosis back in our cell discussion. Mitosis is essentially the division of the what?
The genetic.
Okay. You might just want to say the division of the cell. Yeah, it is. But more specifically, mitosis is actually the division of the nucleus. Does that make sense? Did you get it? Yeah. So it's the division of the nucleus. It's really, mitosis is more about what's happening with the nucleus than anything else. The chromosomes condense, the nucleus breaks down, the chromosomes separate, then the nucleus reforms, right? That's mitosis. Why can't we say then that bacteria go through mitosis?
Because they're
They don't have a nucleus.
Because they don't have a nucleus, right? Remember, bacteria are what kind of cells?
Prokaryotic.
Prokaryotic. So because they officially do not have a nucleus, we can't say that they go through mitosis, since mitosis is really focused on what happens at the nuclear level. They don't have a nucleus. So it doesn't mean they don't have DNA. Of course, that cannot be the case because all living organisms have DNA. So they certainly have DNA. They just don't have that distinct structure called a nucleus. Therefore, they divide by binary fission. Asexual reproduction, that just means it's not really a sharing of genetic composition from two parents, right? So again, the cells divide in half. The information is copied. They divide. Some of these bacterial cells can divide very quickly, right? Not all. Some bacterial cells have been noted, this is really quick, but have been noted to divide every 20 minutes. Think about that. If a bacterial cell can divide every 20 minutes, and we don't have to do the math, we don't have to get too high-tech with the math here, but you can quickly see that even one bacterial cell, over the course of 24 hours, could be hundreds. You see, if it's dividing every 20 minutes, that's quick growth. And that's also why some bacteria, not all of them, some pathogenic bacteria, it just takes a few bacterial cells inside the body. And then 24 to 36 hours, there's hundreds, right? Thousands, perhaps. And that's why some people get sick from these things very quickly, is because the bacterial cells can, some of them again, not all, can replicate very quickly.
So what is the cost of, like, the death of the bacteria?
How fast does a bacterial cell die? Is that what you're asking?
What is the cost of death? Like,
Oh, well, again, many bacterial cells can replicate very quickly, but there's kind of an ultimate limit of replications, and that's because of certain issues. So it's not really an age issue. It's more of a how many times has that bacterial cell actually replicated. That's more of the issue at play. And so bacterial cells do die in the official sense of dying. But because they replicate so quickly, you could also argue that bacterial cells, even though one singular bacterial cell may die, it's that bacterial cell, because it's divided so many times, it's still living. Does that make sense? You see where I'm going with this?
but like when a bacterial cell dividing into two cells, are these two cells brand new cells, or?
They're brand new cells, but they have the same genetic composition as the original cell that they divided from.
So is there like a time where all the bacteria might die?
Like all of them?
Yeah.
So yeah, there's certain issues that might, like in humans, there's certain issues that might produce that effect. For example, if you take an antibiotic, that may kill all the bacteria, and whatever bacteria is causing the problem, right? That may kill them all at once. Is that what you're asking?
Can the same bacteria reproduce like multiple times, or only one?
Yeah, so in essence, yes. So let's say this bacteria right here, maybe 20 minutes later, that's already divided into two. So this is where it gets complicated. Does that bacteria still exist?
Oh.
Does that make sense?
Yeah.
If this one divides into two new ones, does this one still exist after it's divided?
No.
You see where I'm going with this?
Yeah, I didn't think through that.
Yeah, and then those two. So, right. So even though a bacterial cell could actually die officially, because it's replicating so often, it's actually carrying its genetic information to the next one.
Yeah, I didn't think that through.
Okay. So it gets very technical when you say, can they die off? Yes, they can. Actually, a bacterial cell could die, but probably it's still living because it's replicated itself already. Does that make sense?
Yeah.
Okay. This, not too much to copy, and we've talked a lot, the video, right, focused on flagella and that amazing structure, kind of like an outboard motor, right? I'm not going to say much else now because that's what the video did, went through some of those issues with the flagellar structure, and then it got into a lot of those issues with what happened back again in the 90s with Michael Behe and as he proposed irreducible complexity, and that's all very interesting, super interesting to me. And yeah, the bacterial flagella is incredible. But not every bacteria has a flagella, right? I need to point that out here. It's not like every single bacterial cell or every single species of bacteria has flagella. That's not true. Some do, some don't. But the ones that do, of course, they're using this for locomotion purposes. Actually, some cells have multiple flagella. So some bacteria, it's not like they just have one single flagella. Some have several. So it all depends. There's still some diversity here, even in the bacterial world. Yes.
If the cell's flagella is damaged, do they grow back?
And if that one singular flagella is damaged, will it? You know, that's a good question. What's the regeneration potential of a damaged flagella? I'm not sure 100% the answer to that. My suspicion is yes, but I'm not necessarily 100% confident with that. I would actually have to look that up and see. I'm sure it's been a lot of research. You know, what happens when there's a damaged flagella? Does it regenerate? Does it grow back? Yeah.
The picture that like is black and white, is that actually like seen through a telescope?
Yeah, so this is actually electron microscope image of this structure here. So that's actually it. Yeah, in the membrane itself.
So cool.
Yeah. Yeah, and those are proteins. So you say it looks like a motor. It does.
Yeah.
But that's not metal. Right? It's not a metal engine, like metal componentry. Those are protein structures that are making that, but it looks just like an engine. Yeah, it looks just like a motor structure. It's amazing. Yeah.
So we always show like pictures, like either like really bright colors or like black and white, because we can't like show the color on the inside. But are normal cells like, are they actually colored? Like most cells, do they actually have a color?
Most do not. Most cells would not have a distinct coloration.
So what color are they?
Well, let me back up. So the cells that would have a very distinct coloration are those that have pigmentation on purpose. Obviously, plant cells have chloroplasts, so the pigmentation there is green purposefully. Like our body cells, a little bit different. Like in other words, if you just took off a skin cell and put it under the microscope, it would just kind of look. I mean, at the cellular level, it would really look transparent. Same thing. Remember the cheek cells at the beginning of the year?
Yeah.
Remember you had to stain those because otherwise they're pretty much just transparent. Some cells, like muscle cells, they tend to be more red in color.
And like our red blood cells, they're red.
They are, yeah. So depending on the pigmentation, red blood cells, the hemoglobin and the iron and the oxygen that's coming together, that's producing the red tint. So yes, I would say most of the cells in our body are kind of drab to transparent. There's a few that are have distinct colors, but not most.
What determines if a bacteria or a cell has a
It's just all related to the species. Does that species of bacteria need to move? Or is it a species that simply lives in a certain location and has no need to move from place to place? If it's an aquatic bacteria living in like a pond or a watery environment, typically they will have flagella. If it's like a bacteria that just lives or occupies, let's say, the intestines of a human, they don't need flagella. They just live and grow in that location. Does that help? Yeah. Okay. What we got next? So go ahead and go ahead and copy this. This is kind of the 5 essential components here. Just watching the time. Say what? There's some variation beyond this, but every bacterial cell would have to have at least these 5 things. They have to have genetic information. That's the nucleoid. I'll clarify that in just a moment. Ribosomes, which produce, what do ribosomes produce? Proteins, yeah. So in other words, they need proteins, protein structures. Cell membrane, cell wall, that's not surprising. And then there's another structure here called the capsule. We're going to talk about that. In fact, we've got 2 more slides to go today. The next slide, I think, is the capsule explaining that. And then the next one after that, I think, is the nucleoid. And then we'll stop in our discussion at that point. So the statement up the top there, yeah, it's kind of interesting if you think of it. 1950 doesn't sound that old to me. I know it sounds old to you guys. 1950 sounds like it's forever. My father was born in 1949, so yeah, this is my father's lifetime, right? Doesn't sound that old to me. But it wasn't until like 1950s that's when we really started to understand a lot more about bacteria. For the longest time, it was just considered that bacteria were these tiny organisms, pretty basic, didn't have much in their composition. They weren't that complicated. And now, of course, we're understanding a lot more about these guys. So we're realizing they in fact are quite complicated. Okay, let's do the capsule and the nucleoid, and then we'll be done for today in our discussion. So again, bold print, that's really what you need. The capsule, if you look, I know this image is not perfect. The worksheets that I'll give you a little bit later will have this image, exact same image on it, and you'll be able to see this close up. But there's really 3 layers here. Most bacteria have kind of 3 layers. The outermost layer, which is the red tint here, the outermost layer is that capsule. And then underneath that, you have the cell wall, and then eventually the cell membrane. So what is the capsule? Well, it's kind of a jelly-like covering of bacteria cells. And this capsule is quite interesting. It's jelly-like, but sometimes this capsule will produce certain toxins or certain chemicals, which really protect the bacterial cell. Now, it's also what may make us sick. Say, well, what is it actually about the bacteria that might produce sickness, illness? Often it's the chemical that they possess or produce in the capsule that causes the problem. Now, let me explain this word here, because this is, I think, the first time that we come across this term, phagocytosis. In Greek, phago just means, it means to eat or consume. Cyto is the cell, typical cell reference, right? Sys just means process. So if we break this down, it means eating cell process. Here's the point. Bacteria are actually the food sources for a lot of other microscopic organisms. You're going to learn this when we get to protists. There's a lot of protists down there in the watery environments that would love to eat bacterial cells. They love to consume bacterial cells. So bacterial cells actually become a pretty large food component for other cells. Some bacterial cells will actually produce chemicals in their capsule, which, right, not actually, but in essence kind of taste bad to other organisms. And so those other organisms then do not consume it. Does that make sense? So it's really a protection, anti-predation device for some bacterial cells. But also, because they're producing these as an anti-predation, sometimes it does cause problems in other organisms too. But that's what the capsule is. And then also kind of a sticky, slimy, sometimes bacterial will form very thick colonies. And when they form thick colonies, it produces like a slimy covering, a slimy surface. Now that slimy surface is all the capsules that are all together. But that slimy covering is actually thousands or probably more like millions of individual bacterial cells all come together. Does that make sense? Yeah.
So it says that it surrounds some bacterial cells, but I thought a capsule had like all bacteria.
Yeah, so that is true. This is not emphatic all.
Oh.
But how does it say again? Some, yeah, some. So this is not emphatic all. Not all have the capsule.
Okay.
Some bacteria do not have a capsule, which means their outermost layer would be the cell wall itself.
Okay.
But there's a good portion that do. Okay, last thing here, nucleoid. Yeah, and we're going to stop here in our discussion. So once you get this down, there won't be any more notes today. So again, we've already said that a bacterial cell does not have a nucleus. Yeah, that's true. It is a prokaryotic cell. Doesn't mean it doesn't have DNA. It has to. We might even say it has chromosomes, although they're not the typical chromosomes that we would think of in cells like our own. So what happens is this. Bacterial cells have DNA. The DNA seems to hover in a certain region of the bacterial cell, not a nucleus, but a certain region. And that region we just call a nucleoid. So the nucleoid is kind of a region, an area of the bacterial cell where you will typically find the DNA or the bacterial chromosome. So in that sense, then, it's really the same as what we would think of as the DNA of a eukaryotic cell. That is, the DNA controls the genetic production of the cell. It controls aspects of the cell, determines their properties, their functions, and so forth. So not all bacteria are the same at the genetic level. That is certainly true. Some bacteria have different structures that others don't. Some bacteria have different genes that other bacteria don't have. So even at the genetic level, not all bacteria are the same. Actually, bacteria are probably some of the most diverse things on the planet. We just don't think of them very often because, well, we've got our own lives to lead. And why are we thinking about those invisible things that God has created that we don't need to think about, right? I mean, how many of you on a daily basis, unless you're sick, right? But how many of you on a daily basis think about bacteria? Yeah, I'm seeing a lot of hands. That's what I thought. I'm seeing no hands. Most of you are not going to just sit down and ponder and think about bacteria unless you have reason to do so. All right, what I'm going to do, pass out this worksheet. We have about 16, 17 minutes by the time I get this thing out. Let me stop the video that I started for David here.